Imagine the genome as the most important book ever written, containing the complete story of a living thing. For a long time, we could only read it, but not change it. What if you had a magic pen to correct errors? This is the power of gene editing, like a find and replace for DNA. This revolutionary breakthrough could change medicine, agriculture, and our understanding of hereditary diseases. Thanks to new tools like CRISPR, we can now edit the very fabric of life. So what is CRISPR? CRISPR is essentially a gene editing system that nature invented. It was first discovered in bacteria as their immune system, a way to remember its enemies. When a virus attacks, the bacterium uses proteins to snip out a piece of the virus's DNA. Scientists discovered that they could use a protein called Cas9, a molecular scissor. They create a custom RNA guide to match any gene they want to target. They put this guide together with the Cas9 protein and introduce it into a living cell. This is what makes CRISPR so powerful and easy to use in the lab. Or CRISPR has changed biology forever. Before CRISPR, gene editing was messy and difficult. CRISPR made gene editing accessible, cheap, fast, and precise. This new power has enormous implications for our future in medicine and agriculture. With CRISPR, we could potentially fix errors in our DNA directly. Some scientists are even exploring using it to bring back extinct animals. The possibilities seem almost endless. For decades, scientists dreamed of rewriting the code of life to cure diseases caused by DNA mistakes. Early techniques involved inserting new DNA into cells, but these methods were imprecise. You couldn't control where the new DNA went, leading to risks like disrupting existing genes or causing cancer. Later, tools like ZFNs and TALENs offered more precision, targeting specific DNA sequences. However, they were complex, expensive, and required specialized expertise, limiting their widespread use. Science needed a tool that was precise, simple, and cheap. The stage was set for a revolution. The story of CRISPR starts in microbiology, not a high-tech lab. In 1987, Japanese scientists found a strange repeating pattern in bacterial DNA. Over the next decade, other scientists found the same patterns in other bacteria. In 2002, Ruud Janssen named it CRISPR. Later, Francisco Mojica discovered the spacer DNA matched viruses that attack bacteria. He proposed CRISPR was a bacterial immune system, storing viral DNA snippets like mugshots to recognize and destroy future invaders. Understanding that CRISPR was a bacterial immune system was a huge step. But the next question really was, how did it actually work? How did the bacteria use these stored mugshots to fight off an infection? Scientists around the world raced to figure out the mechanics. They discovered that when a bacterium with a CRISPR system detects an invading virus, it quickly makes an RNA copy of the relevant spacer DNA, the mugshot of that specific virus. This RNA molecule is like a wanted poster. It contains the exact code needed to identify the enemy. This was the first piece of the puzzle. The system used an RNA guide to find its target. Now, this RNA guide does not work alone. It teams up with one of the CA's proteins, the ones encoded by genes found next to the CRISPR repeats. The most famous of these is a protein called Cas9. The RNA guide essentially latches onto the Cas9 protein, which acts like a pair of molecular scissors. Together, this RNA protein complex forms a highly effective search and destroy machine. The RNA guide leads the Cas9 protein through the cell, scanning all the DNA it encounters. It is looking for one thing and one thing only, a DNA sequence that perfectly matches the blueprint on the RNA wanted poster. When the complex finds its target, the DNA of the invading virus, it locks on. The guide RNA holds it in place, making sure it is at the exact right spot. Then, the Cas9 protein gets to work. It acts like a precision scissor, making a clean cut through both strands of the virus's DNA. This cut effectively disables the virus, neutralizing the threat and saving the bacterium's life. The beauty of the system is its specificity. The RNA guide ensures that the Cas9 protein only cuts the foreign viral DNA, leaving the bacterium's own genome untouched. It is a simple, elegant, and brutally effective defense mechanism. It was this mechanism that made scientists' eyes light up. They realized that this natural system could be repurposed. If you could create your own artificial guide RNA in the lab, You could theoretically tell the Cas9 protein to cut any DNA sequence you wanted. You would not be limited to targeting viruses. You could target a gene in a mouse, a plant, or even a human cell. All you would need to do is synthesize a short piece of RNA that matched your gene of interest. This insight transformed CRISPR from a curious piece of microbiology into a potentially world-changing technology for editing genomes. The race was on to prove it could work. The final leap from understanding the bacterial system to creating a functional gene editing tool was the result of brilliant work from many scientists, but two teams in particular made the critical breakthrough. One team was led by Jennifer Doudna at the University of California, Berkeley, and Emmanuel Charpentier, who was then at Umeå University in Sweden. They were studying the CRISPR-Cas9 system in a specific bacterium. In a landmark paper published in 2012, they showed exactly how the system worked. They demonstrated that the Cas9 protein could be guided by a single piece of RNA to a specific DNA target and cut it in a test tube. They also made a crucial simplification. In nature, the guide system involves two separate RNA molecules. Doudna and Charpentier figured out how to fuse these two into a single synthetic guide RNA or sgRNA. This made the system much simpler and easier to use. Now, a scientist only needed two components, the Cas9 protein and a single, easy-to-make guide RNA designed for their target. In their paper, they explicitly suggested that this two-part system could be used as a powerful tool for genome editing. They had laid out the blueprint for the revolution, showing the world the essential components and how they could be harnessed. Just a few months later, in early 2013, another research group, led by Feng Zhang at the Broad Institute of MIT and Harvard, published another groundbreaking paper. Zhang and his team were the first to show that the CRISPR-Cas9 system worked not just in a test tube, but inside the cells of more complex organisms, specifically in mouse and human cells. They successfully used the system to cut DNA at precise locations within the genome of these living mammalian cells. This was the ultimate proof that CRISPR was not just a microbiological curiosity, but a viable tool for editing the genes of animals and, by extension, humans. This confirmation that CRISPR worked in eukaryotic cells, the type of cells that make up plants and animals, was the moment the floodgates opened. The scientific community exploded with excitement. The simplicity and effectiveness of the CRISPR-Cas9 system were undeniable. It was the tool everyone had been waiting for. The work of these pioneering scientists, building on decades of earlier discoveries, had provided a technology that was cheap, precise, and accessible to almost everyone. The era of CRISPR had officially begun, and biology would never be the same again. The power to easily rewrite the book of life was finally in our hands. To understand how scientists use CRISPR, let's imagine opening a gene editing toolkit. The first item is the Cas9 protein, the molecular scissors that cut DNA. Next, the guide RNA, or gRNA, acts as the system's GPS. It finds the exact location in the genome where we want to make a change. We also need the living material we want to edit, cells containing the genome. Finally, our toolkit contains instruments and chemicals needed, including methods to get the Cas9 and guide RNA into our target cells. This delivery process is a crucial step. The first step in any CRISPR experiment is to design the guide RNA. This is arguably the most critical part of the process. The goal is to create a guide RNA that is a perfect match for a specific location on the target gene and unique enough that it doesn't accidentally match any other part of the genome. To design a good guide, scientists use powerful computer software to scan the entire gene, looking for all possible target sites. Once the best guide RNA sequence has been chosen, the next step is to physically create it through a process of chemical synthesis. Before using the guide in a real experiment, scientists will often mix the newly synthesized guide RNA with the Cas9 protein in a test tube to confirm it works. After designing and preparing the CRISPR components, the next major challenge is getting them inside the target cell. A cell is surrounded by a protective membrane that acts like a gatekeeper, controlling everything that goes in and out. You can't simply drop the CRISPR system on top of the cells and expect it to work. Scientists have developed several clever methods to breach these defenses and deliver their genetic editing cargo. The choice of method often depends on the type of cell being used and the specific goals of the experiment. Each delivery technique has its own set of advantages and disadvantages. One of the most common methods is called electroporation. This sounds complicated, but the principle is actually pretty simple. The cells are placed in a special liquid between two electrodes. A brief, powerful pulse of electricity is then applied. This electrical jolt temporarily creates tiny pores or holes in the cell membrane. For a short moment, the gate is open. During this window, the Cas9 and guide RNA molecules can slip inside the cell. The pores then quickly close up, trapping the CRISPR machinery inside, where it can get to work on the cell's DNA. It's a highly effective, if somewhat forceful way to deliver the package. Another popular technique uses modified harmless viruses as delivery vehicles. This method, known as viral transduction, takes advantage of what viruses do best, injecting their genetic material into cells. Scientists can remove the harmful parts of a virus and replace them with the genes that code for the Cas9 protein and the guide RNA. When this engineered virus infects the target cell, it injects the CRISPR instructions. The cell's own machinery then reads these instructions and starts producing the Cas9 and guide RNA itself. This method is incredibly efficient, especially for cells that are hard to work with or when editing cells within a living organism. A third, more direct method involves physically injecting the CRISPR components into the cell. This is done using a technique called microinjection, which requires a very fine glass needle and a powerful microscope. The scientist carefully punctures the cell membrane and injects a tiny volume of liquid containing the pre-assembled Cas9 gRNA complex directly into the cell's nucleus, where the DNA is stored. This method is very precise and highly efficient, as you know for certain that the cargo has been delivered. However, it's also very slow and labor-intensive, as each cell must be injected one by one, making it unsuitable for experiments involving millions of cells. Once inside the cell, CRISPR-Cas9 scans the DNA for its precise target sequence. When it finds the match, Cas9 cuts both DNA strands, a double-strand break. The cell rushes to repair this break using one of two pathways. The quick fix, NHEJ, often introduces small errors, disrupting the gene. The precise method, HDR, lets scientists provide a template to rewrite the gene exactly as desired. This moment of repair is where gene editing truly happens. CRISPR's greatest promise is curing genetic diseases at their source. For blood disorders like sickle cell anemia, doctors edit stem cells outside the body, correcting faulty genes in the lab. These repaired cells are returned to the patient, producing healthy blood and potentially curing the disease. This ex vivo approach allows for careful screening before treatment. Early clinical trials show life-changing results, offering hope for permanent cures. The era of treating genetic disease at the DNA level has begun. In 2023, the first CRISPR-based therapy, Casgevy, was approved to treat sickle cell disease and beta thalassemia. Instead of fixing the faulty gene, CRISPR reactivates fetal hemoglobin, bypassing the disease entirely. Patients once plagued by pain are now living free of symptoms. Stories like Victoria Gray's show CRISPR's real-world impact, yet the treatment remains complex and costly, limiting access. The next challenge, making these cures affordable and available to all. CRISPR is revolutionizing agriculture with precise, targeted edits. Scientists create disease-resistant crops, climate-resilient crops, and improve nutrition by tweaking specific genes. Examples include mildew-resistant wheat, longer-lasting tomatoes, and allergen-free peanuts. CRISPR helps crops adapt to drought and salinity, vital for food security. These advances promise healthier food and more reliable harvests. Precision gene editing is shaping the future of farming. CRISPR accelerates animal research and transforms livestock. Scientists model human diseases in animals and create virus-resistant pigs or heat-tolerant cattle. Edits can improve meat quality or reduce allergens in milk. These changes boost productivity and help animals adapt to climate change. Ethical and regulatory questions remain, especially for food animals. Society must balance innovation with safety and transparency. CRISPR's reach extends to conservation and industry. Gene drives could control invasive species or disease-carrying mosquitoes, but ecological risks are high. In industry, engineered microbes produce fuels, medicines, and materials like spider silk or vanilla. CRISPR streamlines biomanufacturing, making it more sustainable. Synthetic biology powered by CRISPR is reshaping how we make and use everyday products. The technology's potential is vast, but so are the responsibilities that come with it. While CRISPR therapies initially targeted single-gene diseases, the future lies in tackling more complex conditions like heart disease, diabetes, and cancer. These result from interactions between multiple genes and environmental factors, making them difficult to treat. Scientists are developing CRISPR tools to modify multiple genes simultaneously, opening new treatment avenues. Imagine targeting multiple genetic weak points contributing to heart disease, adjusting cholesterol, and reducing inflammation in one intervention. In cancer, CRISPR can edit immune cells to target tumors. New precise CRISPR versions offer a safer way to perform complex edits, bringing us closer to effectively treating challenging diseases. CRISPR's talent for finding specific DNA sequences is now being harnessed for diagnostics. The same system can be repurposed to create rapid, highly sensitive, and inexpensive tests for diseases, producing a visible signal when they find their target. This transforms CRISPR into a powerful disease detector. Imagine a simple paper strip test, similar to a home pregnancy test, that could detect a virus like COVID-19 in minutes. It can also be used to find the telltale signs of cancer, allowing for much earlier detection. They can distinguish between different strains of a virus or identify antibiotic-resistant bacteria, allowing doctors to quickly choose the most effective treatment. The simplicity and low cost of CRISPR diagnostics mean they could be deployed globally. CRISPR technology is now being used in de-extinction projects, aiming to bring back species that have long vanished. One famous project aims to resurrect the woolly mammoth by editing the DNA of its closest relative, the Asian elephant. Scientists plan to use CRISPR to match the elephant's DNA to the mammoth's blueprint for cold adaptation genes. Reintroducing these herbivores could restore the grassland ecosystem and combat climate change. However, de-extinction raises ethical and ecological questions about our role as stewards of the planet. While CRISPR's precision is a strength, it's not yet perfect and can make mistakes. One worry is off-target effects, where CRISPR cuts at unintended locations, potentially disrupting vital genes or activating cancer-causing genes. Scientists are refining the technology and developing safer alternatives like base and prime editors. Another concern is the delivery method, often using modified viruses, which could trigger immune reactions. Long-term follow-up studies are crucial. Editing embryos raises concerns about hereditary changes passed to future generations. Altering one gene could have unforeseen consequences. The most contentious ethical debate surrounding CRISPR revolves around human germline editing. This means making heritable genetic changes to human embryos, sperm or eggs, that would be passed down through generations. On one hand, this technology holds the theoretical promise of eradicating devastating hereditary diseases forever. However, this capability puts humanity on a very slippery slope and raises some deep philosophical questions. By altering the human germline, are we crossing a fundamental boundary? They worry that it commodifies human life, treating children as products rather than gifts to be cherished. The discussion quickly moves to the prospect of designer babies and genetic enhancement, creating a new form of social inequality. In 2018, this debate moved from theory to shocking reality when a scientist in China announced he had created the world's first gene-edited babies. This event served as a global wake-up call, highlighting the urgent need for strong international regulations to guide the responsible use of this powerful technology. Beyond ethics, equity and access are immediate concerns. The first CRISPR therapy costs over $2 million, putting it out of reach for many, especially in developing countries. This creates a paradox. A cure exists, but access is limited to the privileged few. This disparity could widen health and lifespan gaps between rich and poor, exacerbating social inequalities. How can we make these treatments affordable and accessible? CRISPR can create nutritious crops, but who will own the seeds? Small farmers may be unable to afford them. Equitable sharing of CRISPR's benefits is a critical challenge. The power of CRISPR extends beyond the human body and into the natural world, raising a whole new set of complex ethical questions. The development of gene drives, a technology designed to spread a specific genetic trait rapidly through an entire wild population, is a prime example. While it could be used to wipe out disease-carrying mosquitoes or control destructive invasive species, it also represents an unprecedented intervention in ecosystems. What would happen if a gene drive accidentally crossed over into a different non-target species? Once released into the wild, a gene drive could be impossible to recall, with the potential to cause irreversible ecological damage on a global scale. The very idea of using CRISPR for de-extinction forces us to reconsider our relationship with nature. While the thought of seeing a woolly mammoth walk the earth again is captivating, it raises difficult questions. Do we have a right to resurrect species, effectively playing the role of creator? And what are our responsibilities to these newly created animals? They would be born into a world that is not their own, into ecosystems that have long since adapted to their absence. Some critics argue that de-extinction is an act of hubris that distracts from saving the thousands of species currently endangered due to human activity. These technologies also challenge our fundamental concepts of what is natural versus unnatural. For thousands of years, the genetic makeup of species was shaped by the slow process of natural selection. Now we have the ability to direct evolution with speed and precision. This blurs the line between nature and technology. When we eat a gene-edited tomato or see a gene-edited animal, are we still interacting with the natural world or with a human invention? These questions don't have easy answers, and different cultures will have very different perspectives on where the boundaries should be drawn, leading to complex societal debates. Ultimately, the awesome power of CRISPR forces us to be more thoughtful and deliberate than ever before. It is not just a tool for scientists in a lab. It is a technology that has the potential to reshape our society, our environment, and our very definition of life. It demands a global conversation involving not just scientists and policymakers, but also ethicists, philosophers, religious leaders, and the public. We must proceed with a profound sense of caution, humility, and a deep respect for the complex systems, both biological and social, that we are now able to change. The future we build with CRISPR will depend entirely on the wisdom we bring to its use. We stand at a remarkable moment. CRISPR has given us a power that was once science fiction. We have found the instruction manual for life, the genome, and in CRISPR, we can finally edit it. This tool represents a fundamental shift in our relationship with the natural world. The ability to change the very code of life is both exhilarating and deeply humbling. It is a power that carries with it a weight of responsibility. The discoveries of the past decade have propelled us into a new era. We are now the stewards of this incredible capability. The potential benefits are immense. The promise of CRISPR is a future with less suffering. With great power comes the need for great wisdom. We have unlocked a door to a new world of possibilities. This new dawn is a dawn of new responsibilities. The decisions we make will shape the future of our species. We must all participate in the conversation about how these tools are used. We are the first generation to hold this power. We have a profound duty to wield it with caution. As we move forward into the CRISPR era, we face profound questions about what it means to be human. Where do we draw the line between curing disease and enhancing traits? Who will have access to these technologies? We must also ask about our relationship with nature. Do we have the wisdom to use this power responsibly, considering the unintended consequences? If we edit the human germline, what kind of future do we want to design, and what values should guide us? This is a conversation about the very soul of our species. The path forward requires a concerted effort from all sectors of society. The first step must be a commitment to robust and transparent science and openly share findings. This commitment to scientific integrity is the bedrock upon which all public trust and responsible policy must be built. We need to create strong, adaptive, and international regulations. We must also invest heavily in public education and engagement. Ultimately, charting our course in the age of CRISPR is a test of our collective wisdom. It depends entirely on the choices we make to shape a future that is brighter for all of us.